ecological environment and climatic conditions. So these are some of the factors that enables the invasive species to spread easily in the new environment. So water hyacinth or Eucornia crassipes is one of the most dangerous invasive species, and it is found in 50 different tropical and subtropical countries of the world. So which demands huge amount of water and which causes a permanent damage on other native plant species. So this water hyacinth is recorded as one of the top 10 worst weeds in the world, and this is extensively and aggressively spreading from time to time. So this affects the biodiversity of organisms, the biodiversity of aquatic species like the water quality, the water trafficking, fishing, and other activities are blocked or affected by water hyacinth. Nowadays, allelopathic studies have get attention for a better weed management. Therefore, there were previous attempts of eradicating or minimizing water hyacinth, like biochemical methods, biological methods, chemical methods, and mechanical methods were some of the methods that were applied for controlling this invasive weed or water hyacinth. But all of these methods were more or less inefficient in a better way controlling of this water hyacinth. Hence, searching for an alternative weed management is currently demanding from the scholars. Therefore, some of plants like Lantana camara and others like this species were reported to have phytotoxicity to water hyacinth, and therefore the allelopathic chemicals as a biological controlling mechanism is a replaceable role in controlling this invasive species. So cypress papyrus are grown with water hyacinth or Eucornia crassipes. So to develop this project, the first observation was made when cypress papyrus was grown with Eucornia crassipes in Lake Tana shore. And then when we are observing this situation, the density and the coverage of water hyacinth was limited, or a kind of scarcity distribution where water hyacinth was grown with this cypress plant species, whereas where water hyacinth found free from this cypress papyrus was highly crowded and abundant. And then we thought that the allelopathic effect of cypress papyrus against water hyacinth has not been investigated so far. And then the aim of this study was to evaluate the allelopathic effect of aqueous and methanolic extracts of this cypress papyrus plants against growth and biomass formation of this invasive plant or water hyacinth with these specific objectives. Materials and methods: plant collection and soil preparation. The cypress papyrus plant was collected from Lake Tana vicinity, and then it washed and allowed for drying at room temperature in order to avoid any other UV harmful and then or damage, and then grinded by electric grinder, and then prepared for further extract. And then similarly, similar size and shape of water hyacinth was collected from the vicinity of the water or from Lake Tana, and then this allowed for growing in pots containing 5 kilogram sandy soil, as you can see from this picture. So this looks like growing of water hyacinth in pots. Experimental design and treatments: about 24 pots, 12 for each treatment. For each extract, for methanolic and for aqueous extract, were filled with soil, and then the experiment was conducted in completely randomized design manner with 3 replicates or replications, and then equal sized and shaped water hyacinth was allowed to grow in each pot. And then treatments were arranged in 4 groups, like treatment 0, the control group, and treatment 1, which receives 10% of the extracts, treatment 2, which plants received 15% of extracts, and then treatment 3, plants grown or water hyacinth grown with 20% of extracts of this alcohol in water. And then preparation and applications of extracts. So the standard solution was prepared by gram to 100 ml, or weight to volume. 1 gram powder was mixed with 10 ml of solvent and ethanol and water separately, according to this recommendation. And then this mixture was mixed for 24 hours and shaken with shaker by 225 revolution per minute, and then kept for 24 hours. And then this mixture was filtered with number 1 Wattman paper, and then the residue was discarded, and then the filtrate was used for further experimentation or experimental purpose. And then the filtrate again, the solvents were evaporated at 95 degrees centigrade for water extract and at 73 degrees centigrade for methanol extraction. And then after separating these solvents, the standard solution was prepared, which considered as 100% concentration, and then kept in refrigerator. And then about 10%, 15%, and 20% concentrations were prepared for both treatments, for both alcohol and water, and then by mixing with 100 ml of distilled water. About 1 milligram of this extracted material was dissolved in 100 ml of distilled water to just prepare 1%, and then to prepare 10%, 10 milligram of the extracts was mixed with 100 ml of distilled water to prepare 15%. 15 milligram of the extracted powder was mixed with this 100 ml of solvents. So this picture shows that this picture shows that mixing and rotating in a shaker, and then this picture shows that when the extract is filtered with Wattman number 1 paper, and then this shows that further evaporating of the solvents according to their temperature. Then Eucornia crassipes plants were grown and acclimatized for some period or for a month, and then with sufficient amount of water, and then extracts were applied for 3 days interval of watering, and then the experiment was extended for 3 months, and then the following morphological and physiological data were collected, like length and number of petals, shoots, and roots were measured and counted every week as recommended by different authors. And the roots length was measured at the end of the experiment after harvesting. And then the following physiological and biochemical data were measured, like chlorophyll content by using this formula, mainly chlorophyll A and chlorophyll B were determined by using this formula at this 663 and 645 absorbance for this chlorophylls. And then chlorophyll fluorescence was also measured by using this portable multi-mode of this equipment, which is available in our laboratory. And then before measuring this chloropluorescence, the leaf was pre-darkened for 13 minutes in order to just capture the full concentration of this, or to get the complete reaction center. And then by using this formula, fluorescence variance or variable fluorescence divided by maximum fluorescence, which gives maximum fluorescence minus the basal or non-variable fluorescence divided by maximum variance. And then biomass was estimated at the end of the experiment by using these procedures. All plants were harvested, the samples, and then finally the root shoot ratio was computed using this formula. And then some of the secondary metabolites or the phytochemicals were identified from the cypress papyrus plants, because after having the effects, let's check what are the secondary metabolites that were responsible for this effect by using this formula, like glycosides was determined by mixing 50 milligram of dry extract with 1 ml of ethanol, and then which is added with 1 ml of water again, and then finally the aqueous sodium hydroxide was added, and then if it gives the yellow color, this was a positive confirmation for the presence of this glycosides, and the same is true for others with these respective methods or protocols. And then results, the effect of this cypress papyrus extract on water hyacinths was significantly affecting these different parameters of the water hyacinths, like the petile lengths and the number of petals and the number of buds or bud formations were affected by both methanolic and aqueous extracts. We can see here that this is a control group, plants or water hyacinths grown without the extract. This one receives 20% of the extracts of methanolic extract. This one, 50% of methanolic extracts. And this one, those water hyacinths grown with 10% of methanolic. And the same is true for the same treatments are applied for aqueous extracts. Here, we can see that the treatment 3 or plants which grown under 20% of methanolic extracts were affected by 50% compared to the control, whereas those plants or water hyacinths grown with the same concentration in aqueous extract were affected by 80% compared to the control in the petile length. So when we come to number of the petals, the methanolic extracts affects by 20% in treatment 3, and it was affected by 73% in aqueous extraction. So we can see that the number of buds, for example, here, the methanolic extract affects under treatment 3, for example, by 68%, whereas under aqueous extract, it was affected by almost 100%, which means no buds were formed from these plants which receive aqueous extracts. So this has significant effect. The effect of cypress papyrus leaf extract on root lengths. So the same significantly, the root lengths were affected by this both the methanolic and the aqueous extracts. Here, the methanolic extract is affected by 62% of under treatment 3, whereas the aqueous extract is affected by 50% here. So the effect on shoot lengths, so both the aqueous and the methanolic extracts are also significantly affecting this shoot lengths. On biomass, both the methanolic and the aqueous extracts also have significantly affecting this biomass formation, but in the case of methanolic extract, the treatments affects or affected by 20% compared to the control, whereas in aqueous extraction, the treatment 3 plants were affected by 34% compared to the control one. And then the effect on pigments, so mainly chlorophyll A and chlorophyll B, as well as the total chlorophyll contents were significantly affected by both extracts. But we can see here that most of the time the aqueous extracts are affecting more the pigments than that of the methanolic extracts. The effect on chlorophyll fluorescence, you can see here that still these methanolic and aqueous extracts are affecting chlorophyll A, chlorophyll B, as well as the total chlorophyll. So this shows that the phytochemical screening of these chemicals, and this the first picture shows that the methanolic extracts, and this the second picture shows that the aqueous extracts of these 6 different secondary metabolites, and the summary is given in table 7. For example, saponin is found both in methanolic and aqueous extracts, whereas tannin is not found, and this anthraquinone is not found in methanolic extract, but it is found or detected in aqueous extracts. So this picture shows that the comparison effect among the extracts. The first, the red pot contains the plants which are grown under aqueous extracts of cypress papyrus, whereas the blue pots contains water hyacinth which grows under the methanolic extracts application. So we can see here that
sorry, Abiyu, your time's up. Would you go please to the conclusion?
All right.
No need to discuss this. So just go to the conclusion.
The conclusion. I'm sorry. Okay. So my conclusion is that extracts of Cyprus papyrus have effects on growth and survival of water hyacinths. So this effect has minimizing at least the expansion of water hyacinths if it's not possible to eradicate. And this study was a new indication to attempt managing invasive species of this Eucornea crassipes or water hyacinths by using Cyprus extracts for alternative weed management in and around Lake Tana. Therefore, it is recommended that further investigation on the phytochemical properties of Cyprus papyrus should be carried out for a better using of this weed management and to have a full information. And then propagating Cyprus papyrus with water hyacinths is a good strategy to control the expansion of this Eucornea crassipes or water hyacinth. So I would like to acknowledge University of Gondar for financial supporting to conduct this stage work, and then Arba Minch University for inviting us to share our experience and for other support. And thank you finally.
Thank you.